Welcome back, biology students. I forgot to hit record during our Zoom today, so I'm going to go through this one more time. So today we're talking about the historical studies related to discovering DNA as our genetic information. Today we know DNA is a genetic material, but back in the early 1900s we did not know this. A matter of fact, majority of the scientific community believed it was proteins because proteins play a role in so many things in our body. The majority of our chemical reactions involve enzymes. Enzymes are proteins. So a lot of the scientific community at the time believed that proteins would turn out to be the genetic information. Today we know that it is indeed DNA. And the question is, how did we get to the point we are today? How do we know what we know? So the first study we look at is Griffith. Frederick Griffith was observing Streptococcus pneumonia in bacteria. He was actually trying to find a vaccine for pneumonia. So I'm taking the approach of the scientific method, allowing us to kind of review that from early in the year and to identify how scientists actually use it in the real world, right? So observations that Griffith has made are those observations related to Streptococcus. He knew that Streptococcus caused pneumonia. He would have done a little bit of research. He discovered that there's two different strains. One is smooth and one is rough. The smooth is smooth because it has a capsule surrounding it, and the capsule itself is physically smooth. He noticed that the smooth strain caused death in mice, and the rough strain, which did not have a capsule, did not cause death in mice. So thinking of a vaccine, remember that's his goal. We often use dead or weakened versions of viruses or non-deadly strains of viruses to make vaccines so that our immune system can recognize that organism in the future, right? So not only does he have this question that he's trying to answer, what can I use for a vaccine against pneumonia, he also has the question, what caused the death? If he can figure that out, he might have a handle on creating a vaccine. So to make that weakened version of the virus, he heat killed the S strain. And sure enough, it did not cause death in mice. So this is a good start. This could be his vaccine, right? Then he added that heat killed version of the S strain to the non-deadly R strain. And when he combined the two, it still caused death in mice. So something went awry here. If the heat strain didn't cause death by itself and the R strain didn't cause death by itself, what the heck happened that caused the combination of those two to become deadly? So Griffith didn't really ever get any further than this. He's left with a question: what caused the transformation? But transformation itself was a really big discovery. Today we use transformation quite often in manufacturing of GMOs, genetic therapy, and other genetic research. So it was a really big discovery. Frederick Griffith was killed in an airstrike during World War I when his lab was blown up, and so he never did get to finish this experiment and to discover the vaccine against pneumonia or what caused the deadly transfer. Today we know and we understand what's causing the transformation. So today we know DNA is our genetic material. We didn't know that back in 1928. We know that bacteria have the ability to take up DNA from their environment. So this would have been the heat-killed bacteria, the DNA broken apart, escapes the bacterium, enters the recipient bacteria here, and it's incorporated in the DNA of this recipient bacteria. So once it's incorporated in this genetic genome, if you will, then this bacteria will transcribe, translate, and produce the proteins associated with that gene, which in this case is that shell, the capsule surrounding the S strain. So this is what caused the R strain to become deadly. So then in 1944, Avery, McLeod, and McCarty were really interested in trying to find out what the transforming factor was. They were quite intrigued with Griffith's work. So they did more research related to Griffith's work and working with Streptococcus pneumonia, and they also worked with infecting the mice. So they're going to do something a little bit different. They're trying to find out what part of that bacteria caused the transformation, caused the other bacteria to become deadly. So they took Griffith's mixture of the heat-treated S strain and the live R strain, and one by one, they used destroying enzymes that would get rid of different components of the bacteria, the protein, the DNA, the carbohydrates, the RNA, and then they looked to see if transformation would occur. So here's the first part of it. So we're using protein-destroying enzymes. So if transformation still occurs when you get rid of the proteins, obviously the proteins are not the genetic carriers. If transformation did not occur after destroying the proteins, you could conclude that the proteins were the transforming factor. So in fact, transformation still occurred. Therefore, proteins could not have been the transforming factor. So they did this with each of the different components. They got rid of the carbohydrates. They got rid of the lipids. The mouse still died, which means transformation still occurred. They got rid of the RNA. They got rid of the proteins. The mouse still died, which means transformation still occurred. When they got rid of the DNA, when they used an enzyme that destroyed the DNA, the mouse still lived. This leads them to conclude that DNA must be the transforming factor. And they knew whatever was being transferred was going to be the genetic code. So this tells them, for the first time, that nucleic acid must be our genetic material. So again, the entire scientific community was believing proteins was the genetic material at the time. So this isn't enough, though it is the first time we were able to make a good claim. It wasn't enough to convince the scientific community. So in comes Hershey and Chase. Hershey and Chase are like, you're off your rocker, Avery. It can't possibly be nucleic acids. It must be proteins. So they actually set out to disprove Avery, McLeod, and McCarty with their work with bacteriophages. Bacteriophages are bacteria that, bacteriophages are viruses that affect bacteria. And so they're going to destroy the bacteria. They're literally bacteria eaters. So a little bit about viruses. Viruses are just nucleic acids wrapped in a protein coat. So if the community is really torn between, is it proteins, is it nucleic acids, proteins, nucleic acids, it's really the best organism you can work with because it's only made of those two things. So we can really isolate one or the other in this case. So they're going to work with this bacteriophage, this virus that destroys bacteria. That's this one here. We can see it has a genetic material on the inside. We know it's genetic material. Back then they just knew it was DNA. And it's wrapped in a protein coat. Hershey is actually from Owasso, Michigan, which we are from Michigan as well, and just down the road from Owasso, so that's kind of exciting. He graduated from Michigan State University, so go green. Their experiment has to do with the bacteriophage being made up of only the protein coat and the DNA core. And if we remember back from chapter 6 when we did organic chemistry, we know that all of the organic molecules we studied contained carbon, hydrogen, oxygen. We knew proteins and DNA, the two we're talking about now, also contained nitrogen. What was different about them was their last element. So proteins also contain sulfur, and DNA also contains phosphorus. So Hershey and Chase are going to take advantage of the sulfur and the DNA. They know that the virus is injecting something into the bacteria. They don't know which. And that within minutes, the bacteria will die, and in its place are viruses. So something has tricked the bacteria into making viruses. So whatever that was being injected is for sure going to be our genetic. So what did the virus transfer? Remember, they were setting out to disprove Avery, McLeod, and McCarty. They said they said proteins were the genetic code, right? So they're predicting that what is being transferred is protein. So their experiment is to use radioactive isotopes. They allow you to trace an element through a chemical reaction or through a process. So radioactive isotope of phosphorus is going to be incorporated where phosphorus was incorporated, that being the DNA. The sulfur is going to be incorporated where the proteins are, which was the shell. So the sulfur was absorbed by the shell and the phosphorus was absorbed by the core. They then infected the bacteria with the virus, and phosphorus was transferred to the bacteria, not sulfur. So remember, the phosphorus was taken up by the DNA. So this is just a visual of that experiment. So the green glowing portion is supposed to be the isotope, the radioactive isotope. Remember, the coat, the outside, was the sulfur. Sulfur was not transferred. So in the end, they could separate the bacteria fragments from the viral fragments, and they did not find that radioactive sulfur in the bacterial fragments. They did a separate set of experiments with radioactive phosphorus, which was taken up by the DNA. In the end, they were able to separate out the bacteria from the viruses, and they found the radioactive isotope of phosphorus was in with the bacterial fragments. So this allowed them to say that DNA was transferred, and we knew whatever was being transferred was going to be the genetic material. So now we can say without a doubt, DNA is the genetic material and not proteins. So way to go, Hershey and Chase. We're going to spend the next couple days going through this historical studies. In class, you have the discovery of the genetic material recap. So take some time to digest these 3 experiments that we talked about today, because tomorrow we're going to talk about another handful of experiments. So here, you're going to summarize Frederick Griffith's experiment. If you need some help, click on the link. There's a video, I believe, there. And then you're going to do Avery, McLeod, and McCarty. And again, you can click on this link. It's an animation. You can click one click at a time through the experiment. And another video will help you to summarize Hershey and Chase if you need. So our goal is to get through 1 through 8 today. Summarize Avery, Hershey, and McLeod, and McCarty's. Summarize Avery, McLeod, and McCarty's, and Hershey and Chase's experiments. And that is as far as we will go today. We will add on to this tomorrow. Okay? So that's all I have for you today. Thanks for listening.